health, mind, body, spirit. Hello, you are watching an interesting video about a way to kill drug-resistant bacteria by alternating two antibiotics. Please read this disclaimer carefully. In a surprising new study, researchers show it is possible to kill drug-resistant bacteria by alternating two antibiotics at doses that would ordinarily boost bacterial resistance and survival when used alone or combined. Researchers found a way to kill drug-resistant bacteria by alternating two antibiotics at doses that would normally be ineffective as a cocktail or on their own. Writing in the journal PLOS Biology, the international team, led by Robert Beardmore, a biosciences professor at the University of Exeter in the UK, describes how carefully devised sequential treatments using antibiotics may help combat the rise of resistant bacteria. Professor Beardmore says they found a complex relationship between dose, density of bacteria population and drug resistance, and, as we demonstrate, it is possible to reduce bacterial load to zero at dosages that are usually said to be sublethal, therefore, are assumed to select for increased drug resistance. Another crucial finding of the study is that the technique may also reduce the risk of bacteria developing resistance to antibiotics, thereby extending their useful lifetime. The researchers decided to carry out the study because for decades research has focused on using drugs as cocktails where the drugs help each other as a synergistic combination. But what if there is also an effect from sequential synergy? Sequential treatment worked with antibiotic doses that singly or combined had no effect. For their investigation, the team used a simple test tube model of an Escherichia coli bacterial infection where the bacteria had some antibiotic resistance genes. They tested the effect of two commonly prescribed antibiotics, erythromycin and doxycycline, on the bacteria in three ways: given singly, given as a combination cocktail and given as a sequential treatment. The results shows that antibiotics given in certain sequential treatments cleared the infection, even when those same drugs given at higher doses singly or combined failed to have any effect. Further tests shows that while sequential treatments did not stop drug resistance mutations in the bacteria altogether, the first drug made the bacteria sensitive to the second drug and thereby reduced the risk of resistance developing. The researchers suggest that the fluctuating environment created by a well-designed sequence of alternating precise doses is what sensitizes the bacteria to concentrations that, in a steady environment, would normally induce drug resistance and extend survival. They note that these findings are still at the experimental stage, and there is still a lot of work to do before any sequential treatments are ready for clinical use. The team expects the findings will trigger a series of studies looking at sequential ways to use antibiotics at doses lower than previously thought possible. The study was funded by the Engineering and Physical Sciences Research Council, EPSRC, the main body for funding research in engineering and the physical sciences in the UK. Professor Beardmore is an EPSRC Leadership Fellow in Mathematical Biosciences. The study was funded by the Engineering and Physical Sciences Research Council, EPSRC, the main body for funding research in engineering and the physical sciences in the UK. Professor Beardmore is an EPSRC Leadership Fellow in Mathematical Biosciences. Meanwhile, we recently learned that a multi-drug resistant intestinal bug is spreading in the US. The authorities say the Shigella bacteria are entering the country in infected travelers and causing a series of outbreaks. Notes and references. Thank you for watching this interesting and informative video.